Let me welcome you all to the G. Burroughs Mitre Lecture and to listen to and honor our Mitre lecturer of today, Dr. Juan Bonifacino. As most of you know, the lectureship was established in 1968 in honor of the first director of laboratories and clinics, and the lecture is presented by an NIH intramural scientist to recognize and appreciate outstanding contributions to biomedical research. So in that vein then, I would like to tell you just for a few minutes about Juan Bonifacino, the head of the Cell Biology and Metabolism Program and the head of the section on intracellular protein trafficking of our institute, the National Institutes of Child Health and Human Development. As a sidetrack, I should point out that in the 9 years that I was scientific director, I'm not sure if this was the first or the second time that I've seen Dr. Bonifacino in a tie and white shirt. As a consequence of that, and suspecting that might happen, I didn't wear a tie today. So to tell you a little bit about Juan, he received his PhD in biochemistry in 1981 from the University of Buenos Aires in Argentina, and in 1982 he migrated to the Bethesda, Washington area, where he came as a postdoctoral fellow with Richard Klausner. In 1997, he became the chief of the Cell Biology and Metabolism branch. He has numerous honors and numerous significant contributions to his discipline. I'll simply point out two of his additional tasks. He's the associate editor of Developmental Cell and is on the editorial board of the Journal of Cell Biology and co-editor of current protocols in cell biology. His lab studies the molecular mechanisms that determine protein localization to different intracellular compartments, with a particular focus on organelles that make up the Endosomal-Lysosomal system. His group found that delivery of transmembrane proteins to these compartments is mediated by an elaborate system of sorting signals and adapter proteins. They discovered new sorting signals and adapter proteins such as AP3, AP4, and GCAs. His research to understand the mechanisms of protein sorting have been key to the elucidation of the pathogenesis of various disorders caused by mutations either in the signals, such as some forms of hypercholesterolemia, or in their adapters, such as in Hermansky-Pudlak syndrome, or by their exploitation by intracellular pathogens such as viruses and bacteria. Recently, they demonstrated that the heterotetrameric AP4 complex plays a role in trafficking and processing of the Alzheimer amyloid precursor protein, and that therefore this may be a potential risk factor for Alzheimer's disease. To summarize his work, I decided to give you a quote from Pasteur in 1871, when he wrote, "There does not exist a category of science to which one can give the name applied science. There are sciences and the application of sciences bound together as the fruit of the tree which bears it." And I think that summarizes Juan Bonifacino. So this afternoon, he's going to talk to us, as was said earlier, with all the answers, sorting it all out, signal-mediated protein trafficking in the endosomal-lysosomal system. Dr. Boniface.
Well, thank you, Owen, for your very kind introduction, and also for your strong support of the work that I am going to present while you were scientific director of NICHD. And in you, I also want to thank the NIH for supporting this basic research into molecular mechanisms of protein trafficking over the years. And I also want to thank the NIH for bestowing on me this honor of being able to present to you the work of my lab in what I consider my scientific home and in front of my scientific family, which doesn't make it any easier. So this talk is going to be about sorting signals, adapters that recognize those signals, and the physiological processes in which they are involved. The talk will consist of two parts. First, I'm going to present a historical perspective on sorting signals and adapters to explain how we came about to study this problem. And in the second part, I'm going to present recent, still unpublished work showing a role for one particular adapter, the AP4 complex, in the sorting of the Alzheimer's disease amyloid precursor protein. As you know very well, the interior of eukaryotic cells is organized into a number of membrane-bound compartments, many of which you will be able to recognize in this electron micrograph of a pituitary cell. Now, each of these compartments has a distinct protein composition which is responsible for the properties of those organelles. So the problem that we and many other people in the field have been interested in studying is how is this heterogeneous distribution of proteins within cells achieved? In other words, how are some proteins targeted to some of these compartments while other proteins are targeted to other compartments? In particular, we have focused on the organelles that make up the Endosomal-Lysosomal system, which comprises various types of endosomes, such as sorting endosomes, recycling endosomes, late endosomes or multivesicular bodies, lysosomes, and a class of organelles that are known as lysosome-related organelles, or LROs, which includes melanosomes, platelet-dense bodies, cytotoxic granules, and others. These organelles are interconnected by vesicular transport pathways, and they are also connected to organelles that feed into endosomes and lysosomes, such as the trans-Golgi network and different domains of the plasma membrane. So there are some proteins then that are specifically localized to some of the organelles in this system. For instance, many of you are familiar with the transferrin receptor, which cycles between the plasma membrane sorting and recycling endosomes, but is not found in other organelles of this pathway, such as lysosomes. In contrast, there are other transmembrane proteins, such as LAMP1, which are mainly localized to lysosomes and late endosomes, but are not found in large quantities in the other organelles of this system. So the question, the fundamental question then in sorting in the endosomal-lysosomal system is how are these proteins targeted to specific compartments within this pathway and within the cell. So already in the 1970s, it was proposed that transmembrane proteins likely had information that told them where to go within the cell. And it later, that information, it became clear that it was contained within the cytosolic domains of those transmembrane proteins in the form of an amino acid sequence. It was then proposed that those signals would be recognized by specific adapter molecules, which were components of protein coats, such as clathrin coats. So in essence, signal-adapter interactions led to the concentration of transmembrane proteins within coated areas of membranes of organelles. And a good example of that are clathrin-coated pits at the plasma membrane, which early on were recognized as a site for endocytosis of some receptors from the plasma membrane, as exemplified in this electron micrograph of LDL particles binding to their receptor and undergoing internalization into cells. Through the work of many people, including Ira Pastan, who's here in the audience, these coated pits were shown to undergo a deeper invagination, eventually leading to the formation of a clathrin-coated intermediate, a carrier, such as clathrin-coated vesicles or other transport carriers that have been described since, which then transported these membrane proteins to the compartment where they reside within the endosomal-lysosomal system. So in essence, what these interactions do is to contribute to the packaging of receptors and other transmembrane proteins within coated transport intermediates. Now, although this model was developed during the 80s, it actually took many years to identify the first signals and the first adapters. And the reasons for that were several. One of them was that the signals were not exactly conserved sequences of amino acids, but degenerate sequences that fit one of several consensus motifs. So all endocytic receptors did not share the exact same sequence of amino acids, but there were specific motifs that were encrypted within the sequence of the cytosolic domains. And in addition, the interactions of those signals with adapters were of relatively low affinity in the micromolar level. So the problem was not really tractable at the time with the methodologies that were available. And it was only in 1990 that Brown and Goldstein reported the identification of the first endocytic signal in the tail of the LDL receptor in a landmark paper that was published in JBC, Go JBC, and which is one of the highest cited papers in this field. So they found that the sequence NPXY, meaning asparagine, proline, X meaning any amino acid, and tyrosine function as an endocytic signal for internalization of the LDL receptor and other members of the LDL receptor family. So it was in 1991, about a year later, that I was offered the opportunity of staying at NICHD and to form my own group as a tenure-track investigator. And so I decided to search for an interesting problem to study as a tenure-track investigator. And I considered the possibility of working on the identification of the adapters that bound to endosomal-lysosomal sorting signals. I wanted to find a new project that was different from the one I had worked on as a postdoctoral fellow in Rick Klausner's lab. And I thought that this was an interesting and potentially very important problem to work on. So I then talked to some friends of mine and some colleagues about what they thought about this idea of trying to work on the identification of adapters. And I remember talking to a senior colleague of mine who listened patiently to my plans and who looked at me and told me, you know, Brown and Goldstein have been working on this problem for years and they have not succeeded, so I don't like your chances of finding the adapters. And as it turned out, this person was right, or at least partly right, because it took many years for anybody to identify the adapters that bind to NPXY signals. It was in 1999 that Jonathan Cooper reported the identification of the disabled proteins, disabled 1 and disabled 2, as adapters for NPXY signals from the LDL receptor. And only in 2001, Helen Hobbs and Jonathan Cohen, using a genetic approach, identified another adapter for the LDL receptor NPXY signal, the protein ARH. So it took 9 to 11 years to find this adapter. So this person was right. If I had chosen to work on the adapters for NPXY signals, I would have been toast, and I wouldn't be here talking to you. So what happened? Well, what happened was that at the time, in 1991, it had already become evident that there were other sorting signals that did not fit the MPXY consensus motif. And there were still no publications, but through the grapevine, one heard that, the transferrin receptor doesn't have an MPXY signal and is still very rapidly internalized. And through the work of several people, including Fred Maxfield, who will be visiting the NIH next week, and Stu Kornfeld, and also some work that we had started to do, it became clear that there was another motif that fit this consensus, YXX bulky hydrophobic, tyrosine 2 residues, that could be any amino acid, and a bulky hydrophobic residue that could also function as an endocytic signal. And this is the signal for which we decided to search the adapter. It took us a couple of years and about 3 postdocs to finally identify the adapter that binds to this type of signal. And it was a postdoc in my lab, Hiroshi Ono, who used what was then a new methodology, the YEAST2 hybrid system, in order to identify proteins that bind to the YXX bulky hydrophobic sequences. And he found that the adapters for those signals were the mu subunits of 3 adapter protein complexes, AP1, AP2, and AP3. By then, it was also becoming clear that there was much greater diversity of endosomal-lysosomal sorting signals than we had anticipated. Because in addition to these NPXY and YXX bulky hydrophobic signals, both of which contained critical tyrosine residues, when you mutated those residues, you inactivated the signals, it had become clear that there were other signals that did not have critical tyrosine residues. And it was actually in our branch, work done by Francois Letourneur and Rick Klausner, that resulted in the identification of another type of signal that did not have tyrosine residues, but instead had 2 critical leucine residues, and which for that reason became known as dileucine-based sorting signals. So we decided to try and identify the adapters that bound to this other type of signal, and this actually turned out to be more challenging, and instead of 3 postdocs, it took 5 postdocs, and probably as many years to fully understand how dileucine signals were identified. And what we found is that these signals were, perhaps surprisingly, recognized by the same adapter protein complexes, AP1, 2, and 3, but instead of binding to the mu subunits, they bound to a combination of 2 subunits. And now that there is a crystal structure for this complex, we know that that is because the binding site straddles the border between 2 of the subunits. Okay, but there was more. In further work that we did, we realized that not all dileucine signals were the same, that there were other dileucine signals which had an acidic residue 3 positions upstream of the first leucine instead of 4 positions upstream. And that was enough to completely change the specificity of recognition of those signals. And in work done by Rosa Portogliano, who is now a tenure track investigator at NHLBI, she found that these dileucine signals were not recognized by the AP complexes, and that was another big surprise. There were even more adapters, but instead were recognized by the VHS domain of 3 other adapter protein complexes that had been discovered in our lab and which were known as GIGA 1, 2, and 3. I'm not done yet. Bear with me. I'm getting to the end of this list of signals. Then a Howard Hughes medical student in my lab, Peter Voorhees, who was in my lab for only 1 year, discovered a different type of signal, acidic clusters, which could also mediate rapid endocytosis of some proteins from the cell surface. And those are found in proteins such as furin, but also in the NEF protein of HIV-1. And actually, that acidic cluster is important for NEF to be able to downregulate CD4 from the cell surface. And it was recently that a postdoctoral fellow and a student in the NIH Cambridge program, Wolf Lindwasser and Riddick Chowdhury, found that at least some of these acidic clusters also bind to a subunit of the AP2 complex, the alpha subunit of AP2. And finally, you are probably all very familiar with the ubiquitination of lysine residues in the cytosolic tails of transmembrane proteins, with functions as a sorting signal for internalization and targeting to the MVV pathway and to lysosomes. This is something that we did relatively little work on, but at least for some of these proteins, again, Rosa Puertollano found that ubiquitinated cytosolic domains are recognized by another domain, the gut domain, of the same 3 gigaproteins that we had discovered before. Okay, so here we have now, like 15 years later, a compendium of signals and adapters that mediate sorting of transmembrane proteins in the endosomal-lysosomal system. You can look at it as a dictionary that allows you to read the language of protein sorting in the cytosolic tails of transmembrane proteins. And in many, though not all cases, the presence of these signals allows you to predict how they are going to traffic within the cell. So where do these interactions take place? Well, they take place at different stages of the endosomal-lysosomal system. Interactions with AP2, ARH, and disable2 take place at the plasma membrane and mediate rapid endocytosis of various endocytic receptors, transporters, adhesion molecules, while interactions with the other adapters take place in intracellular sites, and they mediate different sorting steps between endosomes, lysosomes, and related organelles. Now, all of these machineries are extremely important. They are present in all cells, and mutations, as Owen mentioned, in either the signals or some of these adapters, the recognition molecules, are the cause of various genetic disorders. And years ago, in collaboration with Bill Gall, we showed that mutations in one of the subunits of AP3 were the cause of a pigmentation and bleeding disorder known as the Hermansky-Pudlak syndrome type 2. And in addition, again, as Owen mentioned in his introduction, pathogens hijack many of these interactions in order to enter cells and to replicate within cells. And we've also done some work with funding from IATAP on the mechanisms by which the NEF protein of HIV hijacks AP2 in order to promote rapid endocytosis of the CD4 protein from the surface of cells. Now, there are a lot of interesting stories that I could tell you about each of these adapters and each of these interactions, and we've done work on all of them. However, for the remainder of my talk, I would like to focus on the AP complexes. And in particular, I'm going to show new data on a new AP complex known as AP4. The AP complexes are composed of 4 subunits. They are heterotetramers. They have 2 large subunits, an intermediate size subunit known as mu, and a small subunit known as sigma. These 3 complexes are composed of subunits that are homologous to one another, and they all have the same overall structure of a head with 2 ear-like projections, 2 ear-like domains. This structure has been likened by some to a Mickey Mouse head, except that this is not a very good structural model for the AP complexes, because Mickey Mouse doesn't have these long hinge-like sequences that connect the ears to the head. But it sort of helps you remind you what these complexes look like. Now, these complexes are ubiquitous. They are present in every cell in our body and in all of your favorite cells, T cells, fibroblasts, neurons, hepatocytes. They are present in all cells. They are clathrin associated. They use clathrin as their coat, as their scaffold. And they are recruited to membranes not by interactions with the signals. Those are too weak. But they are recruited to membranes by virtue of interactions with members of the ARF family of GTPases and by specific phosphoinositides, specific lipids on each membrane. And the use of different ARF proteins and phosphoinositides confers a different localization to each of these AP complexes. Now, as I said before, AP complexes recognize two types of signal. Remember that? Tyrosine-based sorting signals, YXX bulky hydrophobic, which we showed bind to the mu subunits of the AP complexes, and also dileucine signals, which bind to a combination of two subunits of the AP complexes. So there are two different binding sites for each of these types of sorting signals. The structural basis of these interactions are now well known from the work of David Owen at the University of Cambridge, who solved the crystal structure of AP2 or its subunits in complexes with these signals. Now, we know quite a bit about these three complexes, and I'm not going to tell you about everything we've done about them. However, a few years ago, another postdoctoral fellow from my lab, Esteban de la Angelica, who is now an associate professor at UCLA, and I actually lifted this picture from his website. He looked younger then. And so he discovered the existence of a fourth adaptor protein complex. And this discovery, the same as the discovery of AP3, which was also discovered by Esteban de la Angelica, was a direct product of the EST sequencing project and the genome sequencing project, because that's how we found proteins that were related to the subunits of the two known complexes, and we then demonstrated, we put all those subunits together into these two complexes. So Esteban discovered AP4, and this complex is different from the other three. It's structurally related, but it's different from the other three in that it's not associated with clathrin. And in addition, after the discovery of AP4, we hypothesized that perhaps AP4 would be able to recognize the same type of sorting signal as the other three complexes. So we tested for interaction of AP4 or its subunits with various tyrosine and dileucine-based sorting signals that we had in the lab, and we failed to demonstrate any interaction. So it was really different from the other three. And eventually, we got frustrated with this complex, and we stopped working on it because we couldn't find any signal that bound to this complex. And a couple of other labs did some work on AP4, but eventually the field forgot about this complex for a few years. And it was not until recently, and a little bit serendipitously, that we finally found a cargo molecule that interacts with AP4 and that is sorted in an AP4-dependent manner. And this is what I would like to tell you about now. So this cargo protein that interacts with AP4 happens to be the Alzheimer's disease amyloid precursor protein, or APP. So how did we find this interaction? Well, it was a little bit by accident. Two postdoctoral fellows in my lab, Patty Burgos and Gonzalo Mardones, who are here in the audience, were actually working on a different project. They were trying to demonstrate interactions of APP, the amyloid precursor protein, with the retromer complex, which is another signal recognition complex on which we have done some work, because the group of Scott Small at Columbia University had found that certain sporadic cases of Alzheimer's disease presented with reduced levels of retromer. And he went on to demonstrate that knockdown of retromer resulted in increased production of the amyloid-beta peptide, which is a pathogenic agent in Alzheimer's disease. So we were interested in determining whether APP interacted with retromer. But we couldn't find those interactions, so we failed, again, to demonstrate any interaction of APP with the retromer. But in the course of that work, Patty and Gonzalo had the idea, it was not my idea, it was their idea, to test for interactions of APP with a bunch of adapters that we had in the lab. And they made the surprising observation that the tail of APP interacted with the mu4 subunit of AP4, but not with the related homologous mu subunits of AP1, 2, and 3. A pattern of interaction that we had never seen before. We had seen a lot of tyrosine signals that interact with mu1, mu2, and mu3, but none that interact with mu4. This was just the opposite. So this was really very interesting, and we decided to investigate this in more detail. And this will show you how we go about conducting this kind of studies. Okay, we now have an interesting interaction. What do we do with it? Well, first of all, we looked at the tail of the amyloid precursor protein, the tail of APP, and we immediately recognized an NPXY signal, NPTY, similar to those that Brown and Goldstein had shown mediates endocytosis of the LDL receptor. And in fact, this NPTY signal has been demonstrated to mediate endocytosis of APP by virtue of interaction with various phosphotyrosine binding domain containing proteins. So we decided to test what residues within the tail of APP interacted with mu4, whether it was this NPXY signal or something else. And I'm going to make a long story short. We conducted an alanine scan mutagenesis of the tail of APP, and this is showing the results from this glutamic acid residue all the way to the C-terminus. And as you can see here, mutation of the NPT of the NPTY signal had absolutely no effect on the interactions with mu4 as determined by the yeast 2 hybrid system. So mu4 was not recognizing the NPTY signal, it was recognizing something else. So what is it that mu4 recognizes? Well, the tyrosine was important, although not absolutely essential, because the binding was greatly decreased, but there was still some residual binding. And then the sequence FFE. So the sequence that is recognized by mu4 is this YKFFE sequence that shares the tyrosine residue with the NPTY sequence, but is distinct from the NPXY sequence. Okay, well, this was great. We could actually define a 5 amino acid sequence that was required for this interaction. Interestingly, this sequence fits the minimal consensus motif YXX bulky hydrophobic. Phenylalanine is a bulky hydrophobic residue. But it differs from other YXX bulky hydrophobic sequences in 3 important respects. The first one is that the tyrosine is important, though not essential for interaction with mu4. That's not the case for the other mu subunits. Second, there is a specific requirement for a phenylalanine at this position, which the other mu subunits don't really care for. And finally, there is a requirement for a 5th residue, a glutamic acid residue. So the sequence that interacts with mu4 is a pentapeptide instead of a tetrapeptide sequence with very unique requirements. So this led us to think that we were in the presence of a new type of interaction because the requirements were quite different. And this would prove to be the case with further analysis. Now, we've used a lot the yeast to hybrid system to discover these interactions and to conduct an initial characterization of the interactions, but eventually we always demonstrate and further characterize these interactions using in vitro binding assays and various physical techniques. And just as an example, I would like to show you the results of isothermal titration calorimetry analysis of the interaction of a peptide containing the YKFFE sequence binding to recombinant mu4, which occurs with an equilibrium dissociation constant of about 30 micromolar, which is in the range of what most of these signal-adaptor interactions are, relatively low affinity interactions. And substitution of the tyrosine and the 2 phenylalanine residues completely abolishes interactions with recombinant mu4 in these in vitro assays. Okay, so now we wanted to understand the basis for the unique requirements for this interaction, and we collaborated with Jim Hurley, as we have been doing for the past 8 to 10 years, on various problems related to recognition of sorting signals, to solve the crystal structure of mu4 in complex with the signal from APP. And it was Gonzalo Mardones, my postdoc, in collaboration with Adriana Rojas, a postdoc in Jim Hurley's lab, who managed to solve the crystal structure of mu4 in complex with this signal. And so what does mu4 look like? What does this complex look like? Well, it's an immunoglobulin-like beta sandwich structure to which the signal binds in an extended conformation, with the key residues, the aromatic residues, being the ones that make the closest contact with the surface of mu4. Now, this structure is similar to that of the related mu2 subunit of AP2 in complex with the tyrosine signal from the EGF receptor, which was solved by David Owen at the University of Cambridge a few years ago. So the overall structure of the mu subunit is the same, as you can appreciate here, and as we rotate these molecules. Very, very similar, but with one very important difference. Look at the position of the signal peptide. It's bound on 2 completely different surfaces that are opposite to one another. So the peptide from APP binds to a completely different binding site on the surface of mu4, explaining why the requirements for these interactions are distinct. The crystal structure also revealed the residues on mu4 that were involved in interactions with the sorting signal, and we mutated several of these residues, and we found that mutation of those residues abolished the interactions in the yeast-2 hybrid system, as well as in vitro binding assays, confirming that the crystal structure explained the interactions that we had observed in biochemically. And just to show you an example, again using isothermal titration calorimetry, mutation of one of these mu4 residues that interacts with one of the phenylalanine sequences in the signal completely abrogates binding to mu4. So this is a real interaction. Now, the fact that it's real doesn't mean that it's not artifactual. There are a lot of very elegant and beautiful artifacts and interactions, in particular when you find them using the yeast-2 hybrid system. So this could have been just a curiosity with no physiological significance. But after many years of working with signals and adapters, this really looked very good to me, and I suspected that it would be physiologically significant. And now I would like to tell you about what we did in order to test for the significance of this interaction. But I have to tell you a little bit about the amyloid precursor protein and its trafficking. So APP is a type 1 integral membrane protein that's found in all cells. It's also ubiquitous. It's everywhere. And in most cells, we don't know what this protein does. However, in neurons, it has been shown to function as a regulator of synapse formation. However, the phenotype of knockout mice, for instance, are very, very subtle. So it's really not critical in and of itself for synapse formation. What this protein is better known for is as a precursor for the amyloid-beta peptide, or A-beta, which is a fragment of 40 to 42 amino acids that comprises part of the transmembrane domain of the amyloid precursor protein. This amyloid-beta peptide is produced within cells and secreted into the extracellular space, where it tends to aggregate to form amyloid plaques, which are a pathogenic hallmark of Alzheimer's disease. And in fact, overproduction of amyloid-beta peptide is thought to be one of the main causes of Alzheimer's disease, both familial as well as sporadic forms of the disease. And somehow, this extracellular amyloid-beta peptide leads to neuronal toxicity, neuronal death, and essentially depletion of neurons from vast areas of the brain. Okay, so we decided to investigate then whether these APP-AP4 interactions were involved in the trafficking of APP and perhaps in the generation of the amyloid-beta peptide. So the first thing that we did was to compare the intracellular distribution of APP relative to that of AP4. And we found that APP, this is the nucleus of a cell, was for the most part found on these scattered vesicular structures that colocalized very well with early endosomal markers. So for the most part, APP was found in endosomes, although there was also a population in the trans-Golgi network. In contrast, AP4 was found to have a much more compact and restricted distribution, which colocalized with markers for the trans-Golgi network, such as TGN46. So one possibility would then be that AP4 would be mediating sorting of APP from the trans-Golgi network, where AP4 is, to endosomes, where APP is localized for the most part at steady state. So we decided to test this hypothesis. So what we did was to mutate the FFE part of the sequence of the signal to AAA, and then compare the distribution of this mutant protein to that of the wild-type protein. And we found that this mutation of the signal resulted in a shift in the distribution of APP from endosomes to the trans-Golgi network, as you can see here, to a much more compact distribution. In addition, knockdown of mu4 instead of mutation of the signal resulted in a similar phenotype of redistribution of APP from a mostly endosomal to a mostly TGN localization. So disrupting this interaction in two different ways resulted in the same phenotype of less endosomal localization and more TGN localization at steady state, suggesting that indeed AP4 is involved in mediating the transport of APP from the TGN to endosomes. Now, the production of the amyloid-beta peptide has already been shown to be tightly linked to the trafficking of the amyloid precursor protein from within cells. And in fact, in familial forms of the disease or in sporadic forms of the disease, in some cases mutations in APP have been shown to lead to overproduction of the amyloid-beta peptide, but there are also mutations in some trafficking factors that similarly result in amyloid-beta peptide overproduction and that predispose individuals to Alzheimer's disease. So we thought that then this relocalization of APP by disruption of the interaction with AP4 would have consequences to the production of the amyloid-beta peptide. So I need to explain to you how the amyloid-beta peptide is processed proteolytically, how APP is processed proteolytically to the amyloid-beta peptide, and how this relates to the trafficking of the molecule. So APP is a type 1 integral membrane protein, but unlike some of your favorite proteins, receptors, transporters, adhesion molecules, that live much of their life within the cells as a transmembrane protein, as an intact protein, this is a protein that is made to be destroyed by proteolytic processing. So at any given time, there is relatively little full-length APP within cells because much of it is being very rapidly proteolyzed by two pathways that are known as the non-amyloidogenic pathway and the amyloidogenic pathway. The non-amyloidogenic pathway, which is the major pathway in most cells, including neurons, fortunately, starts with cleavage by alpha secretases right in the middle of the amyloid-beta peptide sequence, effectively destroying the potential to make the amyloid-beta peptide. So these are good enzymes. It's good to have high alpha secretase activity in your cells, in the brain in particular. And however, there is a bad enzyme, at least in this context, beta secretase, which cleaves at the amino terminus of the amyloid-beta peptide sequence, and which creates a fragment from which the amyloid-beta peptide can be produced. These two cleavages result in the production of a luminal extracellular fragment that is released into the medium, into the extracellular space. And some people believe that this is actually the business end of APP, is the reason for cells having APP, in order to release this extracellular domain that functions as a trophic factor for synapse formation. But these cleavages leave behind two very short transmembrane fragments known as CTF-alpha and CTF-beta. So what does the cell with those transmembrane remnants? So it further cleaves those remnants by another enzyme known as gamma secretase, which cleaves within the transmembrane domain to produce another extracellular fragment that is released into the medium, P3 in the case of CTF-alpha and the amyloid-beta peptide in the case of CTF-beta, and an intracellular cytoplasmic domain fragment that has a short half-life, although some people have shown that this fragment can translocate to the nucleus and regulate the transcription of some genes in a manner analogous to the cytoplasmic domain of Notch. Although a lot of what has been shown in the field of Alzheimer's disease and APP processing is controversial. So this amyloid-beta peptide is then the one that is secreted and forms the amyloid plaques. So where do these cleavage events take place, and how does trafficking of APP regulate the amyloidogenic versus the non-amyloidogenic processing of the protein? Well, it turns out that the exact intracellular sites where these cleavage events occur are really not known with certainty. So in working with the amyloid precursor protein, we will have to work while dealing with all of these uncertainties. Alpha secretase cleavage is believed to occur at the plasma membrane or on the way from the trans-Golgi network to the plasma membrane. And this is probably the best understood of all the cleavages. So this cleavage occurs in the late secretory pathway. Beta secretase is a transmembrane enzyme that cycles between the TGN and endosomes, like many other endopeptidases such as furin and others. And so in principle, cleavage could occur either in endosomes or in the trans-Golgi network. And most recent work by other labs appear to support cleavage happening within endosomes. And finally, gamma secretase is the most confusing and most uncertain of all, mainly because there are very low levels of gamma secretase within cells, and it's very hard, if not impossible, to detect the endogenous protein. But gamma secretase has been proposed to cleave the amyloid beta, the APP, at either the Golgi complex, the plasma membrane, or endosomes, although again, most recent studies favor the cleavage happening within endosomes. Okay, so what happens then with the processing of APP by these secretases when we disrupt the interaction of APP with AP4? So in order to analyze this, we had to obtain a lot of reagents and generate a lot of reagents, because as you might imagine, it's actually quite complex to follow the fate of these different fragments. But eventually we obtained all those tools, and we performed various types of analysis, including pulse chase analysis for the processing of APP. And in this analysis, we compared the processing of wild-type APP with mutant AP3 APP with a mutation in the signal that interacts with AP4. And we found that release of the soluble extracellular fragment into the medium, into the extracellular space, was not affected by mutation of the AP4 interacting signal. So alpha secretase cleavage was not affected. So this was not affected. So if this was not affected, we then expected that the levels of CTF-alpha would also be the same upon disruption of this interaction. However, to our surprise, we found that the levels of CTF-alpha, which is the major CTF, were dramatically reduced upon mutation of the AP4 interacting signal. So this was very puzzling. How could two products of the same enzymatic reaction be present at different levels? How could there be an effect on one but not on the other? So we reasoned then that perhaps it was not the generation of CTF-alpha that was affected by disruption of the interaction with AP4, but it was the turnover of CTF-alpha by gamma secretase that could be enhanced upon disruption of this interaction. So we decided to test that possibility in various ways. First of all, we use an inhibitor of gamma secretase known as DAPT or DAPT, and this is now shown by Western blot analysis. Mutation of the signal decreases the levels of CTF, as you can see here. However, when we incubate these cells in the presence of the gamma secretase inhibitor, we now have higher and equal levels of the CTFs for both the wild type and the mutant protein. Indeed, if we inhibit gamma secretase, we now don't see any differences in the levels of CTF-alpha upon disruption of the interaction. So we then made other predictions. Then the disruption of the interaction should increase the levels of P3 and perhaps also of the amyloid-beta peptide, because we are saying that it seems to be enhancing the activity of gamma secretase. And we found that that was indeed the case when we measured the levels of A-beta and P3 secretion into the extracellular space. In this case, the knockdown of mu4 resulted in a 3 to 4-fold increase in the secretion of both peptides into the extracellular space. So the failure of APP to interact with AP4 is amyloidogenic. It increases the secretion of the amyloid-beta peptide. And a final prediction that we made, and this is really an overkill, but this is something that I like. I really like to overkill the problems, because then we are much more convinced that what we are finding is real. Another prediction was that the levels of the intracellular cytoplasmic domain fragment should also be increased upon disruption of this interaction, and we found that that was indeed the case. Mutation of the signal also increased the levels of this fragment by 3 to 4-fold. And incubation with the gamma secretase activity now prevented the formation of this fragment and resulted in equal levels of the CTF fragments for both the wild type and the mutant protein. And although I am showing you only the results of one of these manipulations, both mutation of the signal and the knockdown of mu4 had the same effect. So these are not indirect effects, but they are directly a direct result of the disruption of the interaction. So to summarize these results, which I think exemplify how we go about identifying sorting signals, adapters, and their roles within cells. So we know that AP4 is associated with the trans-Golgi network, probably with a non-clathrin coat. And if there is a scaffold protein like clathrin, we still don't know what that scaffold protein is, and this is something that we are still investigating in the lab. And we think that AP4 mediates transport of at least a fraction of APP, or more likely the CTFs, from the trans-Golgi network to endosomes, en route to multivesicular bodies and lysosomes for degradation, and we have some evidence for this. So probably the main function of AP4 is to dispose of the CTF fragments by the delivery to endosomes and lysosomes for degradation, so that those CTF fragments don't fall prey to gamma secretase, leading to the production of the amyloid-beta peptide. And if there is no AP4, such as when we mutate the signal or we knock down one of the subunits of AP4, then APP and the CTFs are not delivered to endosomes and lysosomes. Instead, they hang around the trans-Golgi network and the plasma membrane, and now we see enhanced gamma secretase cleavage as evidenced by increases in the levels of P3, amyloid-beta, and the AICD fragments. Now, this has the important implication that localization of APP or the CTF fragments within the trans-Golgi network or on the way from the trans-Golgi network to the plasma membrane actually favors gamma secretase cleavage, which disagrees with what some people have proposed concerning the production of the amyloid-beta peptide in endosomes, although it agrees with what some other people have proposed about generation of amyloid-beta peptide in the Golgi complex. And finally, to put it in words, we have found a new signal-adaptor interaction by which APP and AP4 interact, and this interaction results in sorting of APP, or most likely the CTF fragments, from the trans-Golgi network to endosomes. And this might mediate the disposal of either a fraction of APP or the CTFs by delivery to endosomes and lysosomes. We've also made the surprising observation that interaction with AP4 decreases rather than increases gamma secretase cleavage, which is consistent with cleavage happening in the Golgi complex, as some people, but not others, had shown in the past, or on the way from the trans-Golgi network to the plasma membrane. And this has important implications in terms of approaches to try to diminish the formation of the amyloid-beta peptide. And therefore, we think that AP4 exerts a protective effect which guards against overproduction of amyloid-beta peptide by reducing the availability of the gamma secretase substrates, CTF and APP. And therefore, we think that AP4 should therefore be considered a risk factor, as a potential risk factor for Alzheimer's disease. And as is the case for retromer, for which decreased levels of retromer have been shown to occur in tissues from patients with Alzheimer's disease, we think that it will be of interest to now look at those same tissues for the levels of AP4 in affected areas of the brain, as well as to look for mutations in the genes encoding the subunits of AP4 in samples from Alzheimer's disease patients. So now I want to acknowledge and thank the current members of my lab, some of whom worked on this AP4 project or in other projects. Also, former members of my lab, many of whom contributed to the work that I've told you about today, and also a long list of collaborators. Even I was struck at the number of intramural and extramural scientists with whom we have collaborated over the years. And it's really to all of these people's credit that we have been able to elucidate many of these mechanisms. And I want to end with a couple of quotes that relate to the large number of collaborators that we've had in these projects. The first quote is by G. Burroughs Snyder, after whom this lecture is named. And he said that progress has been most rapid in scientific research when investigators pool their specialized resources and skills on a basis of mutual interest and respect. So really, he really praised the value of multidisciplinary and collaborative science, and I think that what we've done over the past 15 years or so is really a testament to the importance of collaborative work. And the second quote is one that I heard Bob Lefkowitz make, but this quote actually is attributed to Abe Weiler, who was the New York Times entertainment editor and critic, who said that nothing is impossible for the man who doesn't have to do it himself. So it's really to all of these people that I have to be thankful for all this work. Thank you very much for your attention.